Get started. So, welcome everybody to our final of this year's ICNS conference. It's my pleasure and privilege to be introducing Professor Ann Campbell, who's a professor in the Department of Applied Mathematics at the University of Waterloo. So, Ann obtained her PhD in theoretical and applied mechanics from Cornell University, after which she went to the University of Montreal as postdoc. And she had a short stint as an assistant professor at the Concordia University before returning to Waterloo, which is where she is now. In her research, she develops and analyzes mathematical models of neural dynamics. She studies such dynamics on a range of different spatial scales, from single cell all the way up to population level. And a major focus of her work is the role of nonlinearities and time delays in shaping these dynamics. So today, Ann is going to talk to us about modulating synchronization with a slowly varying ionic current. And if you have any questions during the talk, please put them into the chat, and I will pose them to Ann if the time is appropriate. And then non-pressing questions will be reserved for the discussion after the talk. Over to you.
Okay, so can you see my slides okay?
Perfect.
Okay, so I just wanted to thank the organizers for the invitation to speak. It's a great honor to speak here. And actually, as the last speaker, I wanted to, on behalf of all the participants, thank the organizers for a fantastic conference. It's I've enjoyed it very much. I think it's been very well organized, and the time zones worked out really nicely for many different people. So thank you very much for that. Okay, so let me get started. First, I want to thank my collaborators on this project. So Issam Aldarabsa was a postdoc that working with me for a few years, and now he's in the Department of Mathematics and Statistics at University of Manitoba. And he did some of the work on the first part of the talk, and then the second part is work with Victoria Booth at University of Michigan and Chuying Wang at Washington State University. And I want to acknowledge the support of the American Institute of Mathematics. So this work was really instituted when the three of us participated in Research Square. We're only three of us, we were a triangle, but at AIM, and that really helped us to kind of get the work going. And also my usual funding agencies and the university that helped support Issam's postdoc. So just a little bit of background. The work I'm going to be talking about today is related to neuromodulators. So neuromodulators are chemicals that are released by neurons that can modulate the network behavior in a neural network by affecting synaptic transmission or by affecting the intrinsic excitability of neurons. So the neurons can release neuromodulators. They might affect a neuron, or it might affect the synapses. Today we're going to be thinking about the one I'm interested in is acetylcholine, which is thought to play a role in arousal, attention, memory, learning. On a smaller scale, it can affect the frequency and coherence of cortical oscillations. So the word I'm going to think about is where it's modulating the neuron, the effect of a neuron. So here's some very old experimental evidence looking at particular neural cell. I believe it's a pyramidal cell, and this is under control, and this is under the effect of acetylcholine. And what you can see is it's changing the spiking behavior. And we've heard a lot in the last few days, at least a lot of talks I went to, we're talking about spike frequency adaptation, and you can see how this acetylcholine is modulating that spike frequency adaptation. So here's a quite strong adaptation. So the spike frequency adaptation is when the spiking time changes over the course of the period of stimulation, and here you can see it slows down and actually stops. And here there's a change in the interspike interval, but it's not quite as strong. So we go from kind of strong spike frequency adaptation to weak. And so one of the ways it acts many different ways, but one of the ways it acts is through potassium current. So in a particular one, the muscarinic potassium current, the acetylcholine acts in a particular receptor, the muscarinic receptor. So a number of people have studied these effects in neurons. So some of the old work is like this. Some newer work has looked at, so Stifel and looked at phase resetting curves of neurons and how acetylcholine affect that. Prescott looked at resonance effects. So there's many different ways that acetylcholine, we observe that it affects the spiking or excitability behavior of individual neurons. And then at the network level, this is some old work that looked at rhythms in the hippocampus, looking at NCA3 and NCA1. And in this case, they use carbachol, which is basically acts like acetylcholine, mimics acetylcholine effect on the IM current. And so you can see these sort of large rhythms arise, and when they shut down, essentially block the receptors, and the rhythms go away. So, so you can see that this had a very large effect on the network behavior. And more recent, really interesting work, acetylcholine has been associated with changes in rhythms during sleep. So here they're measuring in simultaneously EEG and EMG in rats, but they're also measuring these units are neurons that secrete acetylcholine. So there's a kind of a correlate. This is the wake, slow wave sleep, and this is essentially REM sleep. And there's different, you can see as there's different sort of rhythms happen, you can see also changes happening at the same time as acetylcholine. So that definitely seemed associated with that. But the really neat thing about this study is that they actually showed more than just a correlation, but that the fact that acetylcholine induces the rhythms because they used they were able to use the word's leaving me right now. They used genetically engineered rats where they could turn on the production of acetylcholine by these particular neurons by light. Okay, and so they could turn on the production of acetylcholine, and they could see sort of changes from kind of a stronger rhythm to something kind of smaller afterwards. Okay, and they looked at power, changes as power and things like that. So this is really neat because they could show it wasn't just sort of, they happened at the same time, but it really seemed that the acetylcholine was causing this change in the rhythm. Okay, so getting to like, what were we trying to do with this work? It seems acetylcholine has these different, has these effects, the single cell level, and that they may scale up into this effect at the network level. But they tend to vary more slowly over time. So we were interested in how this slow variation may affect the behavior in networks. In particular, we thought about thinking about synchronization. So whenever you see these kind of large amplitude oscillations coming out of the network level, you think of synchronization. So this is going to be two parts. The first part, I'm going to talk about some work that I did with the just looking at the effect of a static effect of acetylcholine on single cells, and just kind of get a kind of general picture of what it does from a kind of bifurcation analysis point of view. And then the second part, I'll really get into thinking of the slowly varying acetylcholine. Okay, so just to set the scene, so I'm really small. This talk is going to be pretty small, single neurons and small networks. So a standard conductance-based model for neural voltage, neural excitability, such as the Hodgkin-Huxley model, looks something like this, right? So we have an equation for the voltage that's varying in time. So V is the voltage across the cell membrane, right? We have the capacitance, standard and applied current, which as you vary that, it'll change the spiking behavior, the spiking frequency of the neuron. And I've sort of rolled all into this variable here, these ionic currents, which are typically associated with producing the action potential. So in the Hodgkin-Huxley, it would be sodium and potassium and lead current. And then associated with those are gating variables, which typically have the form equations in this form. So if we look at the variation, as I mentioned, of I applied, it's quite well known that as you increase the I applied in those models, it mimics what we see in many neural behavior, that you get oscillations arising, and there's kind of a classic way of classifying how these oscillations arise. So originally done by Hodgkin in 1948, you can see as the oscillations start, this is plotting frequency versus I applied. You could have them sort of come in at zero frequency and gradually change, or you could have them come in with sort of a jump in frequency. So Hodgkin did this work in, I think, crab axons are originally, but then in 1998, Rinzel and Ermentrout looked at how this could be related to bifurcations in models of conductance-based models, as I just mentioned. So the class 1 is typically associated with the saddle node and invariant circle bifurcation, or it can also be with the homoclinic bifurcation. And the class 2 is typically associated with the fold limit cycles and a subcritical Hopf. And there also has been work which we'll be thinking about and talking about how that type of excitability may affect how the neurons synchronize when they're put together in a network. But first I want to mention some other interesting work. So if you choose, so in the last plots I had I applied as my bifurcation parameter. And if I take some other parameter in the model and do what's called a 2-parameter bifurcation diagram, it has been shown in many different models using different things as this different parameters, so such as leak conductance or potassium conductance, different, that you get a kind of, you seem to see the same bifurcation diagram over and over again. And I'll just explain a little bit. So the green lines here are fold bifurcation. So this is where equilibria are coming together and disappearing. This is a cusp point where those two come together. The blue line is a line of Hopf bifurcation. So as we cross here, we get the periodic solutions appearing, and this is double. So this is two code-mentioned 2 points, the cusp point here and the Bogdanov Tawkins point. So where the Hopf comes in and sort of ends at the fold bifurcation. And so this was observed numerically, and it's in Barter and Dave Trement's book, this kind of bifurcation diagram. We see it a lot in various models. And then in 2015, there were 2 really nice papers who looked at, how generic is this for, because I just wrote down a sort of a generic conductance-based model. And they looked at sort of how generic is this bifurcation for an arbitrary conductance-based model if we don't put specific, currents in there, but we have the kind of standard currents in there. And so one of the things we see with this is that if we think of now whatever this parameter is, fixing it down sort of low, we get class 2 type of oscillator as we vary I applied. But if we move it up and we then vary our I applied up here, then we get the class 1 down below and class 2 up above. So varying this up per other parameter, whatever it happens to be, can modify your model from being a class 1 to a class 2 oscillator. So Issam and I were motivated by this work and by what's been observed in the work it's observed in neurons that have the M current, to think about whether this kind of bifurcation, so it has been observed in models with the M current, to see if it's whether it's sort of generic for that. So what we looked at was it's a general conductance-based model. So same things I had before, but now we specifically make sure that there's this IM current in there. So the IM current's a potassium current. It looks something like this. We have this is a constant maximal conductance. There's a gating variable M. Okay, here's this is a typical equation for that, and has a reversal potential VK, which is potassium. And what we'll be thinking about is that the effect of acetylcholine is to downregulate, so turn off this channel. So when acetylcholine goes up, the GM will go down. So we're thinking of this as being another parameter that we want to think about varying in the model. The models for IM, so how do you choose the tau M and the M infinity? They've been fit for various different data for various different types of cells. So the M current occurs in interneurons. It also occurs in hippocampus and cortical pyramidal cells. So it's very, very much all over the place in the brain. So building on the work that I mentioned of Kirst et al. And Pereira et al., what we did, I'm not going to go into details, but we looked at this as a sort of general model and did some basically normal form and bifurcation analysis. So I don't want to go into details. You can show equilibrium points. The key point in getting an equilibrium points is they have the M variable and the WI variable have some specific form coming from their equation. And then we have this function that comes from the right-hand side of the voltage equation. It turns out that's quite important in determining sort of what happens in the model. And so, for instance, you can show that the kind of known properties of these functions and the positive parameters. You can show there's always at least one equilibrium point for any set of parameters. But more interestingly, you can formulate conditions. So these two codimension 2 points that I showed in the sort of generic model that we see in many different neural models, we can formulate conditions on when those will exist, because they seem to be acting as organizing centers for the behavior of these models. So again, lots of sort of math swept under the rug, but you have these details. You can say, if you want to have a Bogdanov-Takens bifurcation point, then you have certain conditions that need to be satisfied. And what we can show is that they can be satisfied by choosing, so we need some parameters to vary in our normal form, by choosing the applied current and the maximal conductance of the M current as those two parameters. So this will give you the Bogdanov-Takens bifurcation point, and similarly, you can formulate conditions for a cusp bifurcation point, and you can show those can be satisfied by varying these two. So essentially, this M current together with the applied current can induce these two codimension 2 points, which give that kind of generic bifurcation structure. And you can go on from this actually and look at codimension 3 points, but I don't want to go into that. So here's some examples. This is the two that I mentioned from before, the cortical parameter cell that Stifel et al. Looked at, and this is the hippocampal parental cell looked at by Olufsen and et al. And all of these plots, I have IAP on this parameter on this axis, and GM, the conductance of the M current, on the vertical axis. And then here's one we just created. So we took the standard inhibitory interneuron of Wang-Buzaki and added the M current, and I think we just took the M current from the Stifel model and maybe just and threw it in there. And so the interesting thing is that in all cases you see this so what the prediction was, right, we should get these two codimension 2 points, the Bogdanov, the cusp, in all cases, and they do organize, and we see the sort of same backbone structure of bifurcations. So I said we had the fold, the homocolytic, and the Hopf. And the implications I mentioned before, but just to remind you, if we're in the single cell, this is just to show you from the Stifel model, right? If GM is below the BT, so we're kind of down here, then as we vary I applied, if I look at, I produce as I cross here, I have a cell node on an invariant circle bifurcation. So this is a one parameter bifurcation of voltage versus IAP, and this is just showing as I vary I apply. So here I get periodic solutions, and this is showing the frequency. So I get a class 1 oscillator bifurcation as I go across here. And then when I get above that Bogdanov-Takens point, I get a class 2 oscillator. And there's actually a fairly complicated sequence of bifurcations that we go through to get to that, but I'm not going to go into that much. But the key point here is that we have sort of this generic bifurcation structure that we see in all of these models with the M current. And then by varying the M current, we can switch the system from a class 1 to a class 2 oscillator. And so this is kind of well known when you think about class 1 and class 2 oscillators. Now what happens when we couple them together? So if we look at just 2 cells, then, and with inhibitory coupling, when we're in less than this BT, so we're in the class 1, we get synchronized behavior. So this is just coupling them together with standard inhibitory synaptic coupling. And if it's large enough, then we get desynchronized. So here we get antiphase behavior. So this is all with the pyramidal cell from the Stifel model, but we see the same thing if we do the other models. And this is kind of a busy slide, but just trying to illustrate, like we see that. So what I'm showing here is as we vary GM, I have 2 coupled cells, and I'm looking at are the cells basically synchronized or not? And we're looking at the phase shift, the phase difference between the 2 cells. So this is excitatory coupling, and this is inhibitory coupling. And these are the 3 example models that we looked at. And they're not exactly the same, but we see very similar bifurcation diagrams. When the GM is low, in the excitatory case, we have antiphase coupling, and in the inhibitory case, we end up with antiphase behavior. In the inhibitory case, they're synchronized or in phase. And then as we change the GM, we get higher. We have some sort of in-between behavior. This is they're synchronized, but the phase difference is somewhere between pi and 0, or half a period and 0. And then eventually we get to synchronized behavior. So in the excitatory coupling, and the inhibitory sort of has the opposite behavior. Not so clear. This is all done with numerics, so maybe not so clear what the bifurcation is happening here. And of course, we see some, for larger things, we see some other different behaviors. So this is the Wong-Buzaki. But the idea is we have this sort of same backbone structure with the two coupled cells when we vary this this conductance of this M current, which again, we're thinking about as something that acetylcholine can affect. Okay, and I think I just have one little, before we move on to the second part, little illustration of this, right? So this is in a network of 10 cells with inhibitory coupling. And so as we sort of expected from the two cells, if GM is small, then things are synchronized. And so if we look at sort of, this is just looking at the average voltage, right? So kind of to a very simple metric of network behavior, then we see this kind of large amplitude oscillations, whereas if GM is large enough, they desynchronize. And if we look at kind of the network average, we see this low amplitude behavior. So something that maybe is not so apparent at the, if we measure something at the network level. So if we compare to that old paper, right, where they had the large amplitude, sort of what we think is synchronous behavior occurring when the acetylcholine was acting, right, then the theory would say that that would mean that it would be due to inhibitory coupling, right? Inhibitory, we get synchronization when the acetylcholine is on and not when it's off. And the experiment, this is sort of retroactive model verification. The experiment actually showed that blocking the inhibitory synapses destroys the rhythm. So it's self-consistent. It does make sense with with what the theory is predicting. Okay, so that's the summary part one, right? Just looking at the static effect of this M current. From a bifurcation point of view, we showed that using the maximal conductance of this M current and the standard I apply as bifurcation parameters, you can get these two codimension 2 points, which really generate the bifurcation structure of the model. And if the BT bifurcation is quite important in terms of switching how oscillations are produced in the model when the I applied current is changed. So depending on whether GM is above or below that BT, we can have class 1 or class 2 oscillations. So the idea would be the acetylcholine could, which decreases GM, can switch the model from class 1 or class 2, from class 2 to class 1 excitability. And as we showed, associated with the switch is we expect a change in synchronization behavior when the two or more of the neurons are coupled together. Okay, so this stuff appeared in a paper in Journal Math Neurosci last year. So the second part, right, now we want to think about, we have that sort of structure and understanding of how acetylcholine works in a static case. We want to think about what happens when it's slowly varying over time. And we're going to focus in on how it affects the synchronization. So in the case when the neurons are oscillating, there's of course lots of other things that you could do, but that was the focus for this work. So we're going to take that model that we had, and now sort of we have a generic model, and now we'll think about coupling the neurons together with, in a network with some synaptic currents. And I think I just have, so this is just sort of standard synaptic gating with a gating variable. And this is what I used in those simulations before with the 10 neurons or 2 neurons. So we have a gating variable, we have a maximal synaptic conductance, we have a reversal potential, which will determine whether it's excitatory or inhibitory, and we have some connectivity matrix. So for most of our analysis, we're actually going to focus on 2 neurons, because that gives us, as we saw with those little simulations I did before, that gives you at least a general idea of what's happening. So because we're thinking about oscillatory neurons, what we're going to do is use phase model theory. So I'm going to try and have lots of references down here, but this is, pretty well-known standard work, right? If I have a system of weakly coupled, and I'm going to focus just on identical for now, oscillators. So if without epsilon is representing the coupling, with no coupling, each neuron would be just oscillating on its own. And I'm going to allow for a parameter in here, because eventually that's going to represent my my GM in my particular current. And for some range of values, each in the uncoupled system has an asymptotically stable periodic solution. Okay, and then we think about what happens when you couple them together. And this theory tells us if the coupling's weak, so epsilon is a small parameter, then you can map these solutions to this phase model. So now we have one variable for each oscillator or each neuron, which just represents the phase, right? So what this model is going to tell us about is the relative phase of these oscillators. So these will not tell us about the destruction of oscillations. We're assuming the oscillations are there and that the coupling doesn't destroy the oscillations, it just modulate, changes it in some way, and so the neurons are synchronized or not. And so this phase model will tell us how that how the coupling affects that synchronization. So we have some frequency here that depends on this parameter, and of course the coupling when we do the mapping also depends on parameter. And you can map out, as beautifully discussed in both of these books, how to actually calculate this. You can map it out, you can do it numerically, and we did that for some models using XVP dot. So now what we're interested in is the parameter not just being fixed, but being slowly varying. Okay, so I'm going to put a delta in there, and the idea is we have sort of two small parameters. We have this, the weak coupling, which gives us in its own a sort of small time scale, and then we have a slowly varying parameter. And so Young and Park in 2016 had a lovely paper where they looked at the case when these two timescales are the same, so delta is equal to epsilon, and beautiful work. And so we kind of built on their work because we were interested in actually even slower timescale, so that when so we'll go to a slow timescale later on, that we still sort of see the effect of this slowly varying parameter. Okay, so when we do that, and I'm again not going to go through the sort of hiding all the math, you can again get a phase model. Here I've put it in terms of the slow time, and we have this, and I've, sorry, forgot the frequency, but the frequency is going to disappear in a minute anyways. The point is you have this, essentially when you go through the work and all the perturbation theory, you can show essentially we have the same coupling function we had before. We have now the slowly varying parameter in there, right? That's going to affect you know, the form of this this function, which again we have to calculate through some integrals and that. But we have now a phase model with a slowly varying effect in it. So what we want to do is think about that and how that affects our solution. So as I said, just to illustrate, I'm going to think about what happens if we have two neurons, because it's really easy to visualize and see what happens. So if we have two neurons and the M current is fixed, the GM is fixed, right? Then we're just in the sort of the first case, we have our frequencies and the coupling functions, and you can show it just depends on when they're identical neurons, depends on just their differences, these H functions. So you can actually formulate a single equation for the phase difference phi in terms of the slow time. And we get this nice little equation where H is an odd function of phi. It depends on the parameter. And you can, as I said, you can calculate this given a particular system. So, okay, yes. So what do we do with that, right? If this is just a one-dimensional equation, so you can find equilibrium points, or you can have your second year differential equation class do this. Calculate the equilibrium points by keeping in mind what's behind all this. These equilibrium points of this one-dimensional equation correspond to phase lock solutions of the original full model. So we have two neurons, right? And this is telling us, and they're oscillating, and these equilibrium points are going to tell us they would correspond to a particular phase lock solutions where they're oscillating with some particular phase difference given by phi star. And they're asymptotically stable if H prime at phi star is bigger than 0. So as I mentioned, you can calculate the H for given a model of your system. And so we did this. This is actually for the stifle model. And you can see compute what the H function looks like, and then we looked statically as we varied the GM, how does that H function change? So you can see, again, it's one-dimensional equation. You can do everything graphically. You can see there's always equilibrium points at 0 and pi. So they always have the in-phase and anti-phase solutions, which just comes down to from the original structure of the model with two identical coupled neurons. But as we vary the GM, the stability changes. This is the excitatory coupling case. So it goes from the antiphase being stable, that's right, antiphase being stable with low GM to in-phase phase being stable with high enough GM. And you can see this kind of smooth change, and in between we have these other equilibrium points that appear and disappear, and that causes where we have a sort of transition between the different states. So I think I have summarized over here. So if the M current is low, GM is so on some value, you get anti-phase behavior. In the middle, there's some other phase-lock behavior. And when it's high, you get in-phase behavior. And you can map this out in a very crude bifurcation diagram if we look at the phase as a function of GM. And this should be reminiscent of those pictures that I showed before using simulations. And now we're doing it just with our phase model. So those are simulations of the full two coupled neurons and just looking at the solution. So they were just showing the stable behavior. And here with the phase model, we can actually map out the solutions and look at their stability. And so we have, again, excitatory coupling, low GM, we have the anti-phase behavior that's stable, and then we have a pitchfork bifurcation, right? Giving rise to actually two other phase-locked, stable phase-locked behaviors with different phase difference between 0 and pi, or pi and 2 pi. And then as you increase GM further, you come back, and if you think about identifying 0 and pi, this is just another pitchfork bifurcation, sort of a reverse pitchfork bifurcation, where we, and then we go, the synchronous behavior becomes stable. So this is a kind of actually a really common bifurcation diagram that's seen when you're varying stability. But we're interested in, of course, what happens if the current's slowly varying, right? So this was our phase model with the slowly varying parameter. So GM is slowly varying, right? Now, instead of being this representing static behavior for different values of GM, now we have to think about slowly moving through this bifurcation diagram. So it's a problem in slow passes through a bifurcation. In fact, if we're going from here to here, or we'll also think about going the other way, because in the system, the acetylcholine goes up and down, and the CGM should be going up and down. Then we have slow passage through two pitchfork bifurcations. So we want to think about what's that going to do in our system. So there's some nice work about this. We took a kind of simple approach that really, this is like really beautiful work that looks at a lot of details. We look at kind of a simple, there's kind of a basic thing that you can see that comes out of it. And we looked at a simple way of expressing that also gives a way of understanding the behavior in a different way, let's say. So if we have these pitchfork bifurcations, if we're close to the pitchfork bifurcation, then we can write down a normal form for that, right? So if X is just whatever your amplitude, in our case, think of it as like, say, the phi, the phase difference in the model, then we could write down this normal form of the pitchfork bifurcation with slowly varying coefficients, right? So if we think of taking that phase model and putting it into the normal form, we expect both of these coefficients to end up being slowly varying. And so if we put that in another slow time, so now we pull this epsilon out front, then we get something like this. This is again, this is something that your second year differential equation students can probably solve, just a Bernoulli equation, so we can write down an exact solution, but we have this parameter in there. And when we write it this way, we can write down the exact solution, and we have this 1 over epsilon, and that allows us to do some asymptotic analysis and try and think a little bit about what happens. So we're thinking about what happens as we pass through the pitchfork bifurcation. So again, I don't want to go into too many details. An important sort of thing here will be as we vary, so we're varying a parameter in time, right? So as we pass through here, this sort of integral of this lambda of S is going to be important. So the bottom line, I'll just show you a couple of results. So you can do asymptotic analysis, and this gives a lot of details, but the bottom line is if you start from the trivial equilibrium and you pass through the pitchfork, so we're going to have to think about going both ways, and also think about subcritical and supercritical pitchfork bifurcations. What happens is if you were doing it statically, you have a static bifurcation point. There's a particular value of lambda where the bifurcation would occur, and we could think about at what time that would occur, right? So there's a time where that occurs. When you slowly vary, and this is seen in slow variation through hot bifurcation is probably more well known, but it's the same idea. Essentially, you get trapped close to that stable equilibrium point, and the bifurcation gets delayed. And so you don't jump, you don't see the transition until a later value of lambda corresponding to a later, and so sort of a later value in time as you're slowly varying through the parameter, the bifurcation diagram. And you can do the same analysis with a subcritical Hopf, and we actually see a very similar thing. So you see this delay in bifurcation, and that's going to be really important when we think about, what's going to happen with the slow variation. Now, what happens if we do the opposite? So we don't always think about this, but we need to think about this in this problem because we're thinking going back and forth, and also because of the structure of the two pitchfork bifurcations and how they occur. What happens if we start on the non-trivial equilibrium and then we go into the pitchfork bifurcation from that direction, as opposed to starting from the trivial solution and branching out? And so you can do the same asymptotic analysis, and what you see, again, I won't worry too much about details, is basically we don't have this bifurcation delay. So the prediction from the asymptotic analysis is that you should just slowly follow those non-trivial equilibrium points into the trivial equilibrium point. Okay, so that was just sort of thinking about the pitchfork bifurcation normal form and with a slow parameter. So if we have, of course, have this phase model, and so what we're thinking of is that if we're close, we can think about what's happening sort of close to the two pitchfork bifurcations that I pointed out. So if we're near the pitchfork bifurcation of the zero equilibrium point, we could do an expansion, basically write this in the normal form, introduce a slow time, and then we have the same setup. And of course, we could do the same thing. So we could do sort of have what's happening near the zero pitchfork bifurcation of the zero equilibrium and the pi, right? So we have both of them. And then try and sort of patch these things together. So we have the predictions of the asymptotic analysis, but then we also have that exact solution for the variation of this. So what I'm going to show you next. Oh yeah, and so this is, so to make this a little bit more concrete, we tried some different things, but you need to think about how these parameters are varying with GM, and then of course, how is GM varying in time? So we took everything as just linear. This is probably not a bad assumption when we're close to the bifurcation point. And here, just took a linear ramp in time. We tried different things, but just for the point of illustration, we'll play with that one. And when you do this and you sort of map through all the stuff, what it shows is that if you think about, what is that, how does the bifurcation delay manifest itself, then the delay, how we're going to not see the bifurcation when we're supposed to, but it's going to occur at a later value of the parameter. And what you can show is that the amount it's delayed, so how far along you'll be before you see the bifurcation, is equal to how far you were when you started, how far away from the bifurcation point, which kind of makes sense because you're sort of spending more time close to this equilibrium point, the stable equilibrium point, and getting stuck there longer. But for this linear stuff, it does not depend, it should not depend on the ramp speed. Okay, okay, and then we can write down the exact solution in that case. Good, okay, so we have about 10 minutes left, which is great. So what I'm showing here is sort of the bottom half of my pitchfork bifurcation. So that's the black line. This is parameter. So this is taking the pitchfork bifurcation normal form. So we just have this trivial solution and then non-trivial solution. And then what we're superimposing on that is the exact solution with this linearly varying ramp in time. And there's two plots here. In the first one, we're varying the starting point. So the green, I come in here at some particular value of the parameter. And then I slowly vary the parameters in time according to that linear ramp. And I see what the exact solution predicts. And I'm changing the starting point. And in this one, I'm changing the ramp speed. And so the asymptotic analysis, right, told us that I'm going to miss the bifurcation point, right? I'm going to follow the trivial solution until some point, and then I'm going to jump up and start following the non-trivial solution. And the prediction was that the jump point should be the distance of the jump point from the bifurcation should be equal to where I started. Okay, and so we see this quite nicely here. As I start further away, I get stuck on longer. But if I keep the starting point the same and change the ramp speed, I shouldn't see any, sorry, I'm doing the wrong thing. That's the next picture. So this is going from the trivial solution on to the non-trivial solution. And this is going the other direction. So when we went the other direction, starting on the non-trivial solution and back, we said that there shouldn't be a jump. So we see a small sort of rounding out at the corner. And we can think of that. We haven't totally analyzed that, but it seems to be sort of a boundary layer phenomena happening there because everything's getting small. But more or less, this follows what the asymptotic analysis. So these are the exact solutions of the normal form following the asymptotic analysis. And then if epsilon gets larger, it sort of qualitatively looks the same, but we get a bigger issue happening here. So you need epsilon obviously small enough for the asymptotic analysis to make sense, but this kind of same qualitative behavior is going on. And we can push this further if we want with those exact solutions and see what's happening. And then this is the case with the varying ramp speed, right? So if we start at the same point and we just vary, so the different colors now we're varying how fast we're passing through this. And everything, of course, is scaled by epsilon. So this is just multiplication of epsilon by difference. So we're seeing 0.8, 1.6, 2.4. So they're all slow, but it's just how slow. And so they should all jump at the same point, and the same thing going back. So that does seem to follow what's predicted by the asymptotic analysis. But if epsilon gets larger, it's interesting. Qualitatively, we still sort of see like they start at the same point, they seem to jump down, but we start to see this sort of spreading out of the different trajectories with the different ramp speeds. And again, here the same thing. So the ramp speed, when the epsilon's not small enough, that it does seem to have an effect, and sort of it causes things to spread out during the jump. So that was that's not something that we predicted from the asymptotic analysis, but we'll see that it actually shows up quite a lot in our simulations of the full system. Okay, so now we'll take a minute to think of some predictions for the full model. So the full model, the ones we took our model of the two neurons and we reduced it to a phase model, and we got this bifurcation diagram for the phase model with these two pitchfork bifurcations. And so if we're starting, and now we're thinking of slowly varying this GM, if I start from high, I'm thinking high to low, we'll start with that. We expect to start on the synchronous solution, right? Prediction is there should be a delay, so a spatial delay before we see the fact that the bifurcations occur, and then it'll jump, and then it sort of follows this, and then goes to the antiphase solution. So that's what I've got written down here. So we start to come along, we expect some delay, some jump, and then to move along there. And how much jump, where we see the jump will depend on sort of where we start. Okay, so now we did this with simulation of the full model. So we've got 10 ODEs, so 2 coupled of these neurons, it works out to be 10 ODEs, right? And then what I've plotted, the little circles here are the predicted bifurcation diagram based on the phase model. And then all these colored ones are looking at taking that model and applying a linear ramp to the GM current. And we start at different values. So we start some particular value and sort of close to our in phase equilibrium point. And we can see here, so I'm decreasing the GM, my solution comes along, the bifurcation point somewhere around here. I missed the bifurcation point, then I jump, and at some point I follow up. But see this, we can definitely see this rounding out here up the corner of the bifurcation there. But we definitely see the effect of the bifurcation delay sort of depending on where I start, my starting value, which is up here for my GM, that affects where my jump occurs. And if I just vary the ramp speed but start at the same point, then I more or less see that they jump at the same point. But again, we start to see the effect of this ramp speed on sort of spreading out these solutions. And again, we see this rounding off. So it's remarkably good, actually, given that we have sort of 10 ODEs and reduced to a phase model, and then we did this analysis of the phase model, and really the analysis was looking at sort of close to these, close to the bifurcation points. So it does kind of predict not too badly what's going on. And then maybe I'll quickly just go, I mean, the other way. So the interesting thing, when if you go the other way, start from low to high, now we're going to start on this equilibrium point. We expect to see a bifurcation delay in this case, so we should miss the bifurcation jump point and jump and then follow along. So you can see already there's some hysteresis that comes depending on whether you're increasing or decreasing GM. And again, here's the bifurcation point, and we see, yes, we miss the bifurcation point, and then we jump down. Sometimes we miss it totally, and actually totally miss this whole branch of which corresponds to these other phase lock solutions. And then we come down. So this is the antiphase going down to the in phase. This is changing the initial GM. So this is sort of the starting point. So we see a little bit of the effect of closer, the sort of the further I start, the longer until I jump. And this is the effect of changing the ramp speed. So we start at always the same value of GM, but we change the ramp speed. And again, you see this sort of, we can call it the waterfall effect, where these different, they start to jump down, but the ramp speed is really having some effect on and how they spread out and where they go down. And it's very interesting in this case, so versus the previous case where we definitely saw these other phase lock solutions that are predicted to be stable, right? And when we decrease the GM, we definitely saw those, whereas when we're increasing, we kind of, due to the bifurcation delay, to totally miss those. And the system only sees the antiphase and the in phase. And just to finish up, because I think I'm just about out of time, we did some work with periodic cycling. And so, as sort of expected from what I just said, you get a hysteresis loop. So if we take the blue curve here and we start, and my bifurcation's point somewhere here, we miss that bifurcation, we jump down, we totally miss what's, the other stable branch of equilibrium points, which corresponds to the other phase lock solution. So we go from antiphase to in phase. But then when we come back, right, so we go a little bit out here, and then when we come back, we stay on that for a long time and then jump up, and then we follow these. So whether or not the GM is increasing or decreasing makes a big difference in what we see in this sort of in between range values. And the same thing here when we're changing, this is sort of changing the starting point and changing the ramp speed. Okay, and I think in the interest of time, I'll skip, but this is talking a little bit about, it seemed to work better in some bifurcation points than the other, right, in terms of how well it matched. We did a number of approximations. We could control for the ramp speed and how we varied the GM, but we couldn't control on when you linearize or you do your expansion about the pitchfork bifurcation, what exactly, and we linearize those depend on the details of the model, right? And so we couldn't, if those are sort of large in some sense, that might throw off, like we're assuming all this stuff is order 1 with respect to our parameter, right? And so that's something that could throw some of this off. But let me just finish up with summarizing. So we considered this neural model as a slowly parameter GM, which changes the excitability and the synchronization property. We had looked at the fixed parameter for phase model with 2 neurons with excitatory. So here in the second half, I just looked at excitatory coupling. You can do the same thing with in phase. So if with the parameter fixed, we have these 2 pitchfork bifurcations that switch from the system from in phase to antiphase. And then we looked at the slowly varying phase model using this exact solution of pitchfork normal form and also some asymptotic analysis. And we were able with the linear ramp to sort of determine the switch point between the different behaviors and this delay in the bifurcation. And it actually was a reasonable prediction of the synchronization behavior of the full system with those sort of one-way or periodic linear ramps. So it definitely predicted these bifurcation delays and these sort of hysteresis loops that we get when we're varying this parameter. So the implication, right, if we start, go back to our original idea, we're thinking about this. If we're slowly varying this acetylcholine, right, we get this switch between synchronous and asynchronous behavior, and what the switch is, whether it's for high or low, depends on whether you have excitatory. So here's the bifurcation diagram for inhibitory. And it does this by inducing a change in excitability property of the individual neurons, right? And so one thing, the variation level of this ACA must be large enough to actually pass through the bifurcation. If it's varying slowly, right? If we saw we could delay the sort of miss the bifurcation point, if we only go to here, it might just come back again. Actually, we did see that in some cases if we didn't sort of vary it enough. So you could totally miss, right? This if it's varying slowly and you wouldn't see any change in synchronization at all. The other thing is that with the slow, if you have a slow periodic, so we increase the GM and then we decrease, then we get a hysteresis loop where we see quite different behavior depending on whether we're increasing or decreasing the acetylcholine and correspondingly increasing or decreasing the conductance on that M current. Okay, so that's all I wanted to say, and thank you very much for your attention.
That's great. Thank you very much for an excellent and very interesting talk. I'll open up the floor to questions. So if you have a question, feel free to unmute yourself or write it in the chat, raise your hand. Priscilla's unmuted herself, so would you like to ask a question, Priscilla?
Were you speaking to me?
Yeah.
Hi. And hello, Suzanne. Wonderful talk. I enjoyed it tremendously. And you didn't mention the word bursting, and I wonder if you're thinking of this without saying so as a bursting model.
I'd have to think about that. Potentially, yeah. Right now, we've just been thinking, but yeah. Yeah, it is potential, right? Where you could go from sort of a synchronized thing and then slowly into like an antiphase type of behavior, or like so interestingly in the system, you might go from like synchronized and then have a burst of asynchrony and then a burst back into synchrony. Yeah.
Yes, and it's interesting to me. You have a jump which corresponds to the delay, which we call delay bifurcation.
Delayed bifurcation, yeah, which is like quite a common phenomenon, right? We just were kind of quantifying it.
And you have the delay going one way, but not the other way.
Yeah, yes. That's quite interesting. It depends which way you go through the bifurcation.
Yes, and I find the same thing is true with a bursting model. If I think of your model as a bursting model, then I get this. Then I get that the asynchrony corresponds to that feature of the bursting model.
Right. So you get again this sort of hysteresis kind of loop, right?
Yes. Yes. Yeah. Yes. Yes. Thank you very much.
And we have a question in the chat from Alexandra. Regarding the use of GM as a bifurcation parameter, how transposable are those results, part one particularly, to other types of slow conductances, e.g. calcium activated currents?
Yeah, so I think, so part one, we just particularly looked at this sort of the structure of the GM current, like the way it was, where it's very simple, right? It just has an activation variable and a reversal potential. So if you had a similar structure, but if you had activation and inactivation variable, it might be a little bit more complicated to do the analysis than point one. Although, again, people see these bifurcation diagrams like all over the place. That two-parameter bifurcation diagram is very prevalent. So I would think it could be done with another. The second part actually, yeah, I mean, once you sort of see that this parameter causes this effect of changing the synchronization behavior, then the sort of analysis part really didn't care much what that actual current was.
Thank you very much for that. Do we have any other questions for Sue-Ann? Jonathan Rubin has his hand up. You want to mute yourself there, Jonathan?
Yeah, sure. What happens if, honestly, I can't remember. I might have asked you this question before because I've seen you talk before. So. I'll ask it again. What happens if the cells themselves are the source of the acetylcholine, right? So that the level is connected to how intensely they're spiking. Have you explored that at all?
That question, but it's a very good. So it's like then you'd have like a feedback, right?
Right.
Where yeah, no, that's a really interesting question, right? So if you then the excitability would induce, so it could be either like a negative feedback where if it gets too excitable, then it shuts off the current or something like that. No, we haven't actually thought about that, and that's really interesting question.
Okay.
And how it would, yeah.
Anybody else have any pressing questions? If not, I had a quick question in relation to, so you have your exact solution for the normal form, and then you have your kind of numerical solution, and you kind of qualitatively compared the two. Did you also kind of do a quantitative? Is there a way to do a quantitative comparison, or are they kind of too far?
Yes, we could. So we in the full simulations of the model, we did, at least in one direction, we could get a pretty good. So the one thing we can quantify is how much bifurcation delay we expect, right? And with the linear ramp and everything, we can quantify that we expect, depending on where we start the perimeter, you know, that determines where it jumps basically. And so we did check that, and that seemed to work out at least in one direction. And that's why we're trying to figure out, because of the, you know, we played around with parameters, making the epsilon small enough, and it seemed to work better in one direction or than the other. And yeah, I was just trying to figure that one out. But yeah, so, but that's the one thing that we can really quantify, right? Because that comes out of the asymptotic analysis, that prediction of how much delay you're going to get. Yeah.
Thank you very much.